Hello and welcome to today's video talking about Mendelian genetics and how traits are inherited from one generation to the next. To start this discussion, I've put up this nice little picture of my family here that we took a couple weeks ago. We went to the pumpkin patch for Halloween. And there's me and my wife Kendra and our three kids, Vivian and Elliot and Blake. And then up in front here's my mom and dad. And genetics was always one of my favorite topics in school, mainly because I remembered hearing things growing up like, oh, you have your dad's nose, or you have your mom's eyes, or you have your dad's hair, or lack thereof. And I always wondered how that worked. Like, why do I have some traits from my dad and some traits from my mom? And why does it seem kind of imbalanced that I get more traits from one than the other? And some of them do seem to kind of mix together. And I was always curious about that. And so learning about genetics really taught me a lot about how we do inherit these traits from our parents. And so first I want to talk about the history of studying inheritance and kind of the dominant thoughts leading up to what we now know. And there were two kind of main ideas of how traits were inherited. The first was this idea of blending inheritance, that whatever traits your parents had, your traits would be blended versions of those. So if your dad was really tall and your mom was really short, you'd be kind of medium height. If your dad had black hair and your mom had blonde hair, you'd have somewhere in between, some sort of brownish colored hair. But all traits were just blending of the two versions from your parents. There's also this idea of what we call particulate inheritance, which says that each determinant or each physical form had a physically distinct nature. So each, what we now call gene, had its own physical particle that after fertilization remained intact. So even if I had black hair, I might still have that particulate or that particle from my mom for blonde hair, even though it's not being shown. And this is what we found to be more correct in more of the situations as we learn more about genetics. There are some cases where blending inheritance is kind of what's happening, but more of it leans towards the particulate inheritance. So the first person to really study all of this was Gregor Mendel. He was a monk who lived in Austria in the mid to late 1800s, and he was famous for studying pea plants. They were really easy to do genetics experiments with because they had a relatively short lifespan. He could grow a lot of generations in a short period of time. They had easily observable traits like flower color and seed shape and things like that. And he could easily control which ones pollinated which. Pea plants have the ability to self-pollinate, but what Mendel would do is cut off the stamen, which is the pollen-producing part of the plant, and then he could control which plants received that pollen and really have tight control over which plants were being pollinated. And he made a lot of really, really interesting discoveries along the way. So he came up with this idea of how we all have different characters. And a character in this case is an observable physical feature. And so in pea plants, one of the characters he looked at was flower color. And for that character, there were different traits, which is to say a particular form of a trait, or a particular form of a character. And in this case, the two options were white or purple. The flower color would be the character, and then the form of it would be the trait that those plants had. And the plants that Mendel used in particular are what we call true breeding, which is another way of saying, purebred. So kind of like purebred dogs. If I have a purebred German Shepherd and I breed it with another purebred German Shepherd, they're not going to have Chihuahua puppies. They're going to have German Shepherd puppies. So with true breeding plants, if you had a plant that's true breeding for white flowers and you bred it with another plant that's true breeding for white flowers, the only color flowers they can have are white. And so Mendel's experiments, which have been recreated many, many times over the years, basically looked something like this. So the starting true breeding plants that he had were what we referred to as the P generation, otherwise known as the parental generation. And so here he had a true breeding round plant and he crossed it with a true breeding wrinkled seed plant. So he took the pollen from this one and used it to pollinate these flowers. And then the seeds that were produced are what he called the F1 generation. And the F stands for filial, which refers to the family. And so in this F1 generation, what he noticed was all of the seeds produced were round. So they all matched up with this one up here. None of them had that wrinkled trait. But when he took some of these round seeds and self-pollinated them, what he found was in the F2 generation, now you had some round seeds, but you also had some wrinkled seeds. And they tended to follow this 3-to-1 ratio. For every 3 round seeds, there was 1 wrinkled seed. So 75% of the seeds were round and about 25% were wrinkled. And that really led him to ask a lot of questions. And he started wondering if other traits followed the same pattern. And so he actually looked at 7 different traits: the shape of the seed, the color of the seed, the color of the flowers, the shape of the pods, the color of the pods, the location of the flowers, whether they were mid-branch or at the end of the branches, and whether the stems were tall or whether they were short. And he found that in every case, if he crossed true breeding plants for these different traits, there was always one that was more dominant. If we crossed round and wrinkled, you would always get round. If you crossed purple and white, you always got purple. If you crossed axial with terminal, you always got axial flowers. And so those traits that kind of won out every time were what he referred to as dominant. And he noticed that every time he did this in that F2 generation, that ratio was almost always 3-to-1, or very close to 2.96-to-1, 3.01-to-1. You can see that as the numbers got bigger, they got closer and closer to that 3-to-1 ratio. And so he kind of expanded on that particulate idea, and he proposed that there were genes, which he didn't use that term at the time, but he proposed that there were these determinants that occur in pairs, and during meiosis, these pairs separate from each other, and they produce what are called haploid cells. And again, haploid only have 1 copy of each gene, and those haploid cells that are produced are what we call gametes. The rest of the cells in our body are diploid, meaning they have 2 copies of every gene. And if you have 2 copies, you can have different what we call alleles. So if there's alleles would be kind of the new word when Mendel used the word trait, so different forms of a gene. If you have 2 of the same allele, we call that being homozygous. So that's another term for true breeding or purebred. If you have 2 different alleles, you're what we call heterozygous, meaning you have 2 different forms of that trait. Whether you're homozygous or heterozygous, what's contained in your genetic code is what we refer to as your genotype. That describes what's in your actual genetic makeup. And then that genotype influences your phenotype, which is your physical characteristics. So these were all things that Mendel kind of put together in his studies of the pea plants. And this all together led him to the law of segregation, which says that when any individual produces gametes, the 2 copies of a gene separate so that each gamete receives only 1 copy. And this was modeled very successfully using something that you're all very familiar with, which is a Punnett square. And using this Punnett square, if we use the capital letter R to denote our homozygous dominant and lowercase r for our recessive trait, he found that you could make a very strong prediction. These capital R gametes would separate from each other, and the lowercase r gametes would separate from each other. And when they would recombine after fertilization, all of the offspring have a heterozygous genotype, but they have a dominant phenotype. They're all round. But if he took these heterozygotes and crossed them together, again, our alleles separate during gamete formation, and when they recombine during fertilization, now we have that 3-to-1 phenotype ratio of round to wrinkled, but we know that we also have a 1-to-2-to-1 ratio of genotypes. So for every 1 homozygous dominant, you get 2 heterozygous, and you get 1 homozygous recessive. And so the more he did this, the more he saw that these trends continued and the numbers all kind of matched up. From there, he took it to this idea of, well, what if we look at 2 traits at the same time? So in this case, he was looking at the seed color and the seed shape. So we know that round is dominant to wrinkled, and yellow is dominant to green. And so he took true breeding round yellow plants and crossed them with true breeding wrinkled green seeds. And what he found here was that all of the offspring showed both dominant phenotypes. And he predicted that they had inherited one copy of each from the parents after what he learned from his first tests. And so the question that he asked was, for these offspring that are now heterozygous for both traits, heterozygous for both traits, do these alleles always go together? So would the dominant allele always go together with the other dominant allele? Would the recessive always go with the other recessive? His thought was that it didn't matter. They could assort themselves independently. And so he predicted that there were 4 different possible allele combinations. He could get big R, big Y, big R, little r, big Y, and little r, little y. And so to test that, he crossed plants. And so if we put those genotypes on here, and same over here, what he found was that you still had, now you had a mix again of all of the different phenotypes. And in this case, he found there was a 9 to 3 to 3 to 1 ratio of the different phenotypes. So for every 9 round and wrinkled, there would be round and yellow, there would be 3 round and green, 3 wrinkled and yellow, and 1 round or wrinkled and green. And the more he did this, the more it followed these trends. And again, this led to his second law, which is the law of independent assortment, that says alleles of different genes assort independently of one another during gamete formation. So what that means is during meiosis, if you look at this drawing here, we'll say that the red stands for dominant, the purple stands for recessive, the yellow stands for dominant, and the green stands for recessive. Just because in one round of meiosis, the chromosomes line up this way, so we've got big R, big Y, little y. In another round of meiosis, they could all go the other way. So the dominant don't always go together, and the recessive don't always go together. You can also use rules of probability to predict what's going to happen in a genetic cross. So Punnett squares are really good because they show us the predictions, and then you can take those numbers and use them to predict what's going to happen in a future case. So if you look at the example of a coin toss, if we say that tails is heads is like the dominant allele and then tails is like the recessive allele, and I flip 2 coins, there's 2 possible choices. Heads or tails, heads or tails. And so if I want to know the chances of getting a heads and a heads, it's 1/2 times 1/2 or 1/4. Or if I want to know the odds of getting a homozygous dominant, it's 1/2 times 1/2. And then same thing here. If I want to know the chance of getting a tails and a heads, it's 1/2 times 1/2 or 1/4. And you can take these numbers and use them to predict the composition of a population. And we'll actually, as we move on in this unit, talk about doing some ways to analyze whether or not a population is following these models. And the last thing that we're going to talk about today is how you can track these traits through generations. So pedigree is a family tree that shows the occurrence of a particular trait. And these are the symbols that are common for all pedigrees. So you can see circles are always represented, females are always represented with circles, males are always represented with squares. If they're shaded in, that means they're affected by a trait. And if they're half shaded in, it means they're heterozygous, which means they're unaffected, but they do have a copy of that particular trait. If there's a single line between a circle and a square, that means they have mated. And if there's a double line, that refers to mating between relatives, so inbreeding. And we'll come back to pedigrees a lot more, but I just want to show you a couple quick tricks for determining whether a trait is dominant or recessive based on a pedigree. So this particular pedigree shows a dominant trait. And there's two real ways that you can see that. Number one, any individual that's affected has at least one parent that is also affected. So here you see there's 3 of them, but anybody who's affected also has an affected parent. And then the parents that have multiple children, roughly half of their kids also show that trait. I just realized I'd spelled that wrong. So roughly half of the kids are going to show that trait. If it's a recessive trait, it's actually a little bit easier to determine with a pedigree. If you look here, you've got a parent that's unaffected and another parent that's unaffected, yet they have one of their children that is affected. And so the only way you can have a child or an offspring that carries the trait when neither of their parents do is if that trait's recessive. You'll also notice a lot of times that these are the traits that tend to skip generations. So it might show up here, it might not, and then it skips a generation and shows up again down here. And so that's kind of your slam dunk for knowing whether or not a trait is recessive, is if the affected person has parents who are not affected. So that's where we're going to leave off for today. We'll pick up the next time with some non-Mendelian genetics, so some traits that don't follow purely dominant versus recessive patterns. But for today, that's it. So hope that was helpful.